Okay, so today I kind of wanted to go over a quick overview of enzymes and how they facilitate these chemical reactions that otherwise would be somewhat impossible to occur. So to begin, you have to remember that there are 20 amino acids and that each of those 20 has a different R group. So the basic structure of an amino acid doesn't change. It's got like a carboxyl, NH group, that's fine. So we can just label those always as constant. But what sticks out of one of these bonds is the R group, and this can change. Some R groups, for example, so if we have like a collection here, and remember proteins are made of all these, just in like a small sequence. So for example, some of these are neutral, like they're just a carbon and another hydrogen, so nothing. Some of these have inherently negative charges on them, while others have more positive charges. Some of the others have more hydrophobic or hydrophilic interactions, so that can facilitate where the enzyme works. Sometimes there are small just OH groups that can facilitate bonding, hydrogen bonding in some cases. So when you string all these together, you get this collection of kind of what can form binding sites and structures of these proteins, and some of these proteins turn out to be enzymes. So when you see the visualized protein, it's a lot of like this, and a lot of like squiggle lines and things like this. And then sometimes there's like a second protein in here that makes the full enzyme. And oftentimes, in the case of enzymes, there are these pockets. And in these pockets are where the reaction can occur, because the R groups of, say, these little amino acids right here, let's say this reaction needs to facilitate something where a substrate needs to come in, and it's a very positive substrate. So to bind that, these R groups, they need to be that have that negative charge around there, so that this will be attracted to here. The other case can be true too. You could have a negative be attracted to the positive. In any case, the enzyme pocket is essentially facilitated by these specific R groups in the structure. And remember, the proteins get all this information from the DNA ultimately, but they are so conserved and evolved that the structure ultimately forms this pocket under the right temperature and the right pH. Enzymes can kind of break apart if it's too hot, and they won't function correctly if there's too many H ions, as we saw, those hydrogen ions. So in the case of our reaction that we're still going to take a look at in lab, so say that our blue is the oxalacetate, and the red is that NADH. And remember, this isn't a chemical, this is like a coenzyme type of like protein that facilitates the transfer of the electrons, hydrogens. But for this purpose, essentially these are just bouncing all around. And although there's a lot of them interacting with each other, they are just kind of randomly going every which way. Sometimes this reaction could occur if they ran into each other, but the chances of that are really small. And that's why this reaction has to be facilitated by an enzyme, because you can't, in a free environment, you can't just force these two things to randomly occur together, because chemicals are naturally just going all over the place. However, our enzyme, malate dehydrogenase, it has how it's formed, with all the little protein squiggles. So these are all amino acids. Get some structure in there. Essentially, there are R groups that, say in these amino acids in the pocket, that perfectly interact with oxalacetate. And they also, after the, say, oxalacetate binds, there's a small conformational change that opens up R groups that NADH can bind to. Now these two sources are together, and they're forced together and that reaction is forced to occur. And ultimately, what you get is the malate. So it's still pretty similar to oxalacetase. We'll throw some blue in there. But now you've got a finished product. And just as well, the NAD plus comes out. So this is what we were seeing. This is our finished product. And it was facilitated by these two interacting in this specific enzyme pocket, because they would never have just run into each other. We need this reaction to keep going all the time, and random interactions just don't do the job. That's why enzymes facilitate these important reactions by bringing them in with specific amino acids in their pocket. This can be applied to cancer a lot of times too, because a lot of our inhibitors, for example, say this wasn't malate dehydrogenase, say it was an oncogene, a lot of inhibitors that we design, they actually mimic the target of these enzymes, and they bind this pocket and they stay there, and they won't be catalyzed, nothing happens, and then the entire enzyme's dead. And so that's a targeted therapy for cancer proteins.